We know that the genome is our instruction book for life. Prokaryotes have similar genomes, while eukaryotes have very large genomes. So again, the difference between our prokaryotic and eukaryotic genomes is basically just a few things. The size, all right? Prokaryotes are going to be smaller. They're not as big as us, right? Again, the coding capacity. The genes are continuous. What's another word for non-coding DNA? Anybody know what another word for non-coding DNA? Junk. I don't like that word. We don't know yet. How about that? Instead of junk, let's say we don't know yet what they do. So there's repeating sections of DNA that they found code for proteins. And then there's sections that they don't know what they are for. And they call that junk. I don't think that's just because you don't know doesn't mean that it's junk. So prokaryotes have a higher percentage of operons, which are linked genes, and prokaryotes contain plasmids. And they're the circular DNA that is not in your chromosome. So they have that circular DNA that is responsible for the very quick evolution of prokaryotes. Again, eukaryotes are larger, and they have a large amount of non-coding DNA. They've only found 1.5% codes for proteins. And that these introns, or these non-coding parts, are separate those coding parts. And what's interesting is they say that 45% of the human genome is repeated sequences. They don't really know what that is for. You can have either tandem repeats, which is repeating right next to each other, or interspersed, where it's separated. So again, back to the basics. The DNA will be unwound, and in transcription, so DNA found in the nucleus, right, is replicated. It can make more of itself, or it can be transcribed, made upon with to RNA. Then RNA, through the process of translation, is made into our proteins. And this, again, is the central dogma of biochemistry. Got to put that chemistry in there, right? That's what we're saying happens. But I want you to tell me a little bit about, going from DNA to RNA, and then how do the three RNAs work? There's more than 3 RNAs, but we're not going to talk much about them. But again, RNA is, again, a polymer of a nucleotide with that phosphodiester bond. But instead of deoxyribose, it just uses ribose. And instead of T, thymine, we're going to use U, uracil. And so the pair is TC in DNA, and then it's going to be UC in RNA. RNA is single-stranded instead of the double-stranded DNA. Makes it harder to work with. Anybody ever work with RNA? Yeah. RNA doesn't have that strength because what is it lacking? It's lacking the double helix, but what is it? I said it's stabilized, the DNA stabilized by the what between the 2 bases? The hydrogen bonds. That really stabilizes it. Where the RNA doesn't have that. It's only a single strand, so it's going to fall apart easily. I know that a couple years ago there was talk, the way biotech drugs are made now is that we grow the protein in a Chinese hamster ovary cell, and then we inject people with that purified protein. What if we injected people with RNA and had them make the protein? Then you wouldn't have to worry about Chinese hamster ovary viruses. or DNA getting in there, right? But I don't know if that's going to work because RNA is so hard to work with. But there's a whole institute at Albany called the RNA Institute. Isn't that crazy? A whole institute. Not just a class, not just a lab, an institute based on RNA research. I don't know, does anybody know about making drugs from RNA? No? That would be really neat. All right, so so I think I've beaten into you that RNA is hard to work with. But again, RNA, there's three main types: transfer, ribosomal, and messenger. There's others that if you go into research, you would get to know these. We're not going to know these we're not going to talk about these. But there's small interfering and micro, right? And they have various roles. But let's talk about the roles of the three main types. Transfer RNA. I got to spend some time talking about the structure of transfer RNA to my 8th grade daughter, so I'm hoping that you guys have heard this before. What are these? These are not lines. They should be dashes standing for hydrogen bonding, right? It looks like a clover leaf, and where you have the 3' and the 5' end of your nucleic acid coming together. They're held together in this clover leaf section. Where do we see the amino acid that's on our transfer RNA? We see it up here, right? And then what's down here on this second section? The anticodon, right? So it carries the amino acid here, and this hydrogen bonding. The ribosomal RNA just makes up our ribosome. Ribosomes are mostly rRNA and some protein in there. There's two different sizes, two subunits, one larger than the other. And you can use analytical centrifugation, which basically centrifuge, you know that solid things go to the bottom, liquid things, less dense things go to the top, and we can actually do something where you push it through like a cushion of sugar, and where things will separate from centrifugation based on its size. So you can look at sizes of things that are intact through analytical centrifugation. And I just like saying it's expressed in Spegberg units. That's another neat word here. Messenger RNA is what is going to carry that genetic information from the DNA in the nucleus to the ribosomes. And it's short-lived. Single-stranded, and it's synthesized directly from the DNA. It's a complementary strand, synthesized. We unwind the DNA, starting from the 3' end, and make our RNA on that. So let's look at transcription. In the RNA polymerase, in our nucleus, the RNA polymerase unwinds our DNA, and this is what happens here, and then our messenger RNA is formed. There's intake holes in our RNA polymerase that takes our nucleotides in. And this is just one of the coolest executions on our show all semester long. Here's our DNA.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene.
Unwinding it.
It's unzipping the double helix and copying one of the two strands.
The intake.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
Messenger RNA.
The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene.
Make our codons, right?
The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Sort of cool. Translation is now we're going to take that messenger RNA, go to ribosomes. Where are they? On the rough ER. Rough because of ribosomes. And let's watch how translation works.
Translation is the process by which the information contained in messenger RNA is used to direct the synthesis of a polypeptide. This information is carried in the sequence of bases on the messenger RNA molecule. The polypeptide can be assembled once RNA binds to a ribosome. Ribosomes consist of large and small subunits. The large subunit has two binding sites for transfer RNA, the P and A sites. Transfer RNA carries an amino acid, which will be incorporated into the polypeptide. Its anticodon is a triplet complementary to the codon on messenger RNA that specifies that particular amino acid. A ribosome assembles on the start codon AUG. Transfer RNA with the anticodon UAC and carrying the amino acid methionine binds to the codon. The transfer RNA is in the P site of the ribosome. The A site is available for a second transfer RNA with an anticodon complementary to the second messenger RNA triplet. The amino acid carried by the second transfer RNA binds to the methionine. The first transfer RNA leaves the P site and the second transfer RNA moves there, still bound to messenger RNA. This brings the third messenger RNA codon to the now empty A site, and the appropriate transfer RNA can bind to it. The third amino acid is added to the chain, and translocation occurs once again. This process of polypeptide chain elongation continues until a stop codon is reached. A release factor binds to the A site. It carries no amino acid, but facilitates release of the polypeptide and the messenger RNA from the ribosome.
I love how they just fall away. So I think in your biology classes, you probably have seen this before, but I'm hoping through this class, you've gotten a different appreciation for what's going on here. That it's not just that this does this and this does this, but why is it happening? Right? Why is that bond being formed? How is it being formed in the active site of an enzyme? Why do we lose the attraction of one once it's formed, and then the other one once it is formed? It's all about those interactions between the molecules. Changes in pH change those interactions, can make a change in the shape of your protein. So by knowing that general chemistry 2 and 1, knowing those intermolecular forces, knowing why things happen, I think now I want you to, when you're learning more about biochemistry, keep those basic concepts in your brain about why these things are happening, not just what's happening.